Welcome children. We are discussing a very important chapter, that is Biomolecules, Chapter 9 of class 11th Biology. I have already told you that this chapter is very important, not only from the examination point of view, its understanding is also helpful for understanding of some of the chapters of class 12th. So in our previous session, we have already discussed about what are biomolecules, and then we discussed one of the biomolecule, that is protein. Today in this session, we will discuss two other biomolecules, that is carbohydrates and lipids. So here I want to tell you children that in your textbook, NCERT textbook, in a very haphazard way, the things are given, but I have arranged everything systematically for your better understanding. So let us start our discussion. The first biomolecule that we are going to discuss today is carbohydrates. The carbohydrates are the aldehyde or ketone derivatives of polyhydric alcohol. So what is the meaning of this? I will tell you by showing the picture. Okay. So before that we should know what that was the definition of carbohydrate. But what actually carbohydrate? If a layman, if we will ask a layman, then how he will explain the carbohydrate? The carbohydrate is pronounced as carbohydrates. These are the sugars that provide the body with energy. So we get energy from protein, fat, as well as from carbohydrate. But the immediate source of food energy is the carbohydrate. And this carbohydrate composed of organic compounds like carbon, hydrogen and oxygen. And the carbohydrates are the main source of energy in a living body. That's why they are known as energy giving food. Some examples of carbohydrates are sugars, candy, sweets, ice cream, sodas, milk, rice, grains, breads, cereals, chips, cakes, cookies, biscuit, pizza, pasta, fruit and vegetables. So in almost all food we get carbohydrate. The oxidation of carbohydrate during respiration releases energy which is stored in the form of ATP, and this ATP is utilized by our body cells or tissues whenever required. And this is very important point, last point children, about this carbohydrate. 1 gram of carbohydrate is about 4.1 kilocalorie of energy when oxidized in the body tissue, and this is called calorific value of carbohydrate. So if in exam what is the calorific value of carbohydrate is asked, then you should write it is 4.1. In some book you will get 4.2. Okay, 4.1 or 4.2 kilocalorie. Now I have told you that this carbohydrate is made up of carbon, hydrogen and oxygen. And what is the proportion of carbon, hydrogen and oxygen is present in a carbohydrate? So this is the ratio. So this is, here I have taken glucose. So here you can see that C6H12O6. So the carbon, hydrogen and oxygen are present in 1:2:1 ratio. And the monomer of a carbohydrate is called sugar. The monomer means the single unit or the units which are joined together to form a carbohydrate, long chain of carbohydrate, is called sugar. So here I have shown you the most common type of sugar, that is glucose. So this is the structure of glucose, where you can see only the presence of hydrogen, carbon and oxygen. Now this is the classification of a carbohydrate. The carbohydrates are classified as monosaccharides when only one sugar molecule is present. That means only one monomer is present. Then disaccharides means two sugar molecule or two monomers are present. Oligosaccharide means 2 to 10 sugar molecules or monomers are present. Polysaccharide means 10 or more than 10 sugar molecules are joined together to form the polysaccharide. And here are the examples of monosaccharide, disaccharide, oligosaccharide and polysaccharide. So monosaccharides, the common examples are glucose, fructose, galactose. Disaccharides are sucrose, lactose and maltose. Oligosaccharides are raffinose and stachyose. Polysaccharides are starch, glycogen and cellulose. Now we will see the structure of mono, di and polysaccharides. So here you can see only one sugar molecule is present. So this is glucose. It is a monosaccharide. Similarly, here you can see there are one glucose and one fructose is attached. So these two are the monomers which are attached with one another and they form the sucrose. Okay, so sucrose is formed by the linkage of two monomers, that is glucose and fructose. Okay, so this is a disaccharide. Similarly, polysaccharide means when it is more than 10 or more than 10 sugar molecules are joined together to form a compound, then that is called, form a carbohydrate, that is called polysaccharide. Now, in the definition I have told you that the carbohydrates are the aldehyde or ketone derivatives of polyhydric alcohol. So from here I will explain you what is exactly the meaning of the definition. So the aldehyde and ketone are the functional groups present in the organic compound. The aldehyde, the functional group aldehyde is written as CHO and the functional group ketone is written as C-double-bond-O. This you have to remember. Now, on the basis of the functional group, the carbohydrates are of two types. One is aldolase, that means those carbohydrates which are having CHO, that means the aldehyde as the functional group. Okay, that means the aldehyde derivative of polyhydric alcohol. So here let me explain you. These are the aldolase, that means they are having aldehyde as the functional group. So here you can see CHO is the aldehyde group. Okay, so this is the functional group. Now aldehyde derivative of polyhydric alcohol. Polyhydric alcohol means what? So here you can see there are two carbon atom. This is the functional group. You leave this one. Then see there are two carbon atoms to which hydrogen atom is attached and alcohol group is, hydroxyl group is attached. That means it is a polyhydric alcohol which is having more than one alcohol group and more than one hydrogen. Okay. Similarly, here you can see if you leave this CHO, that is the functional group, then this is a polyhydric alcohol having many alcohol or hydroxyl group and many hydrogen. Okay, so similarly here also this is a polyhydric alcohol having aldehyde as functional group. So this is also a polyhydric alcohol but having a aldehyde as a functional group. So this group of carbohydrate which are having the aldehyde as the functional group are known as aldoles. So the examples are glyceraldehyde, then ribose, glucose, galactose. Similarly, now come to ketose. Ketose means those carbohydrates or those sugars which are having the ketone group, that is C double bond O group as the functional group. So here you can see that it is a polyhydric alcohol, that is if you leave this CO group, that is ketone group, then these two carbon atoms are having two OH group and two hydrogen group. So it is polyhydric alcohol which is attached to a ketone group. So a ketone group is attached to a polyhydric alcohol. Okay, so that's why they are known as ketoses. And the examples are dihydroxyacetone, ribulose, fructose. So many other examples are also there, but these are the common type of aldoles and ketoses. Now, let me tell you what is the linkage present in carbohydrate. Very important, children. In protein, what is the linkage? Peptide linkage or peptide bond is present. Similarly, in carbohydrate, the linkage that is present between the monomer or the sugar unit is called glycosidic linkage. So more often it is asked in the exam to show or to represent how the glycosidic linkage is formed. So here I am going to show you the formation of glycosidic linkage. So this is a glucose molecule. This is another glucose molecule. Okay. So you have not studied about the isomer. So I am not discussing this alpha and D glucose, beta and D glucose. So that will be confusing you. In chemistry you will study about that. So simply you just remember that this is a glucose molecule and this is another glucose molecule. In glucose, how many carbon atoms are there? I have shown you this formula, that is C6H12O6. Now let us see one of the glucose molecule which is having C6H12O6. Let us see. So these that is vertical parts, that is your apex, are having the corners are having the carbon. So how many carbons are there? 1, 2, 3, 4, 5 and 6. 6 carbon atoms are there. So this is the first carbon atom here, then second carbon atom, third carbon atom, fourth carbon atom, fifth carbon atom, then sixth carbon atom is written here. So there are 6 carbon atoms, so C6. Now how H12? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 12 hydrogen, then O6. O, 1, 2, 3, 4, 5, 6. 6 O. So C6H12O6 is the formula of a glucose molecule. So this glucose molecule, in the glucose molecule, the carbon atoms may arranged in the linear form, but when the glucose molecule is forming the bond, then it is in a ringed structure. So this is the ringed structure of a glucose molecule. This is another glucose molecule. So this is the first carbon atom and this is the fourth carbon atom. And to this carbon atom, as I have shown you previously, that to each carbon atom, one hydroxyl group and one hydrogen is attached. Okay. So here also one hydrogen and one hydroxyl group is attached. Now what happens? the linkage that will be formed is taking place between the first carbon atom and the fourth carbon atom. Okay, so now let us see how the linkage is formed. So here what is this? OH, hydroxyl group is there, and here H. So this H and OH together removed as water. Okay, so if you will combine this, then it will be H2O. So now this H2O is released. Then they are linked. So here you can see what is left? O is left. Okay, so here you can see how the bond is formed after the removal of water. The only O is left. So this bond is a glycosidic linkage, and since it is taking place between the first carbon atom and the fourth carbon atom, so it is called 1-4- glycosidic linkage. Very important, children. If you will write simply glycosidic linkage, what type of linkage is present in carbohydrate? If you are writing glycosidic linkage, then it is okay. But if it is asked that by which linkage the monomers or sugar unit in a carbohydrate are linked linearly in a straight chain, then you have to mention the position of carbon atom, that is 1-4- glycosidic linkage. Okay, I hope it is clear to you. So here two glucose molecules are joined together, linked together by glycosidic linkage, and they formed a disaccharide. That means consisting of two sugar units. And what is that? That is maltose. Then come to polysaccharide. So here I am showing you how polysaccharide is formed. So this is the example of starch. Later on we will be discussing in details about starch. So here you can see that this is one glucose molecule, this is another glucose molecule, this is another glucose molecule. So here how the linkage is formed, you can see this is the first carbon atom, fourth carbon atom, again first carbon atom, fourth carbon atom. So they form a straight chain. In a straight chain, the glucose or the sugar units are joined by 1-4- glycosidic linkage. Now we'll discuss about the polysaccharide, because in your syllabus the detailed account of polysaccharides is there. So I will also discuss the different types of polysaccharides in details. The polysaccharides are often referred to as complex carbohydrate because it is made up of more than 10 sugar units which may be arranged in a straight chain or in a branched chain, or they may be forming a coil structure or helical structure. The large polysaccharides are insoluble in water. And what is the function of carbohydrate, that is polysaccharide? For example, starch and glycogen allows the cell to use them for storage of food. You know that in plant the starch is stored, the food is stored in the form of starch. In case of animals, the food is stored in the form of glycogen. In fungi also, the food is stored in the form of glycogen. Then chitin is a type of polysaccharide which is used to make the exoskeleton of arthropod. The insect's exoskeleton is made up of chitin. Then cellulose is a strong polysaccharide that is present in the cell wall of a plant cell. Then cellulose is not easily broken down by most of the animals, including human beings. It can be digested by the ruminants, that you know. Now we'll discuss the classification of polysaccharides. The polysaccharides are basically classified into two types, that is homo and hetero polysaccharides. Now what is homopolysaccharide? The homopolysaccharide contains only a single type of monomeric units. That means suppose we are taking starch. then that starch is composed of only glucose units. Okay, and heteropolysaccharides means the polysaccharides that contains two or more different kinds of monomeric units, like peptidoglycan, which contains different types of sugar units. They are not containing a single type of sugar unit. Again, the homopolysaccharide may be, so here in the picture you can see that in a homopolysaccharide only one type of sugar unit is present. So it is shown by a single color. So this homopolysaccharide may be unbranched or branched. That means they can be linear or they may have branches. Okay. Then heteropolysaccharides consisting of different types of sugar units. So here the different types of sugar units are shown by different colors. So there they may also be unbranched or branched. So here you can see that this polysaccharide, heteropolysaccharide, consisting of two types of sugar units. But here you can see three types of sugar units shown by three different colors and they are branched. Okay, and the example of homopolysaccharide I have told you that glycogen, starch, cellulose, these are all the homopolysaccharides. And the heteropolysaccharide is peptidoglycan, that is the constituent of the cell wall of bacteria. The polysaccharides, as I have told you, that they consist of 10 monosaccharide units or more than that. So I have already told you some of the examples. So here more examples I have given. The monosaccharide, that is homopolysaccharides, are starch, glycogen, dextrin, cellulose, inulin and chitin. The heteropolysaccharides are glucosaminoglycan and glycoproteins, peptidoglycans. Now, so here this is one of the most important thing you have to know, children. There are different types of polysaccharides, but in your textbook the three most important and very common type of polysaccharides are explained. So I will also discuss these three types of polysaccharides, which are very important in the living system, and I will discuss it in details. So what are these three polysaccharides? One is cellulose, I have already told you where it is present abundantly. Then starch, it is the stored food material in plant. Glycogen is the stored food material in animals. Now, so the source I have already told you, cellulose is there in the plant. Then amylose and amylopectin. So the starch can be divided into two types, that is amylose and amylopectin. So when the starch is having only, that is 1 dash 4 dash bond, that means only straight chains are there. Okay, only straight chains are there, then that is called amylose. And when they are having branch, then they are called amylopectin. Okay, so the cellulose and starch are present in the plant, but glycogens are present in the animals and fungi. Now, what are the monomers? The monomers in cellulose are glucose. In starch also the monomers are glucose. In glycogen also the monomers are glucose. Then bond, what type of bonds are present? In cellulose only straight chain is there, and I have already told you that in straight chain 1 dash 4 dash glycosidic linkage is there. So in cellulose only 1 dash 4 dash glycosidic linkage is there. In starch also the 1 dash 4 dash glycosidic linkage is there in case of amylose. But in case of amylopectin, since branches are there, so these branches are formed by 1 dash 6 dash glycosidic linkage. That means the side chain is formed. So here this is the straight chain glucose and this is the branch. So here what happens to the 6th carbon atom, the first carbon atom of other glucose linked. So this is 1-6' glycosidic linkage. So since amylopectin is having straight chain as well as branch chain, so they have 1-4' as well as 1-6' glycosidic linkage. Then glycogen, here also it is profusely branched. So here also 1-4' and 1-6' glycosidic linkages are present. Then here from the picture it is very much clear to you that in cellulose there is no branches, in amylose there is no branches, but the starch type, that is amylopectin, the branches are there. Okay, and approximately per 20 subunits one branch is there. Then glycogen, it is more profusely branched. If you compare the structure of starch and the glycogen, the structure of glycogen is more complicated because in every top 10 subunit the branch is there. So it is more profusely branched and more complex than the starch. Okay, so these are the steps of cellulose, amylose and amylopectin and glycogen are given. Now we'll discuss these three types of polysaccharides in details. Now first one is cellulose. It is the organic compound belonging to the category of polysaccharide and it is made up of glucose subunit. It is found in bacteria and plant cells. It is present in the cell wall of plant cell. And what is the function of cellulose? It provides structure and strength to the plant cell, and the cellulose finds a great importance in the industry, and the cellulose is made up of thousands of glucose subunits. I am not discussing about D-glucose. The glucose subunit in the cellulose are linked by 1-4' glycosidic bond. I have already told you the cellulose is unbranched and the glucose units are arranged in a linear pattern. Okay, and they may also they have as building blocks like amino sugars and chemically modified sugars like glucosamine and acetyl galactosamine, etc. So here you can see the structure of glucose. So this is the leaf. In the leaf the cells are there. The cells are having the cell wall. The cellulose microfibrils are forming the plant cell wall. So this is the structure of the microfibril. So each fibril is highlighted here. So each microfibril of cellulose is a straight chain of cellulose. So this is a straight chain of cellulose. Okay. So here you can see the cellulose consists of only glucose, only one type of subunit, and these subunits are joined by 1-4' glycosidic linkage because they form the linear chain. Now what are the unique properties of cellulose and why this unique property is there? Because the cellulose has a very unique structure, so they have very unique properties. And what are those properties? The cellulose is the most abundant carbohydrate present in the nature. Very, very important. It is insoluble in water. Cellulose is a crystalline solid having a white powdery appearance. Okay. It has high tensile strength. That's why it provides shape to the plant cell, protects the plant shell, and it is possible due to the hydrogen bond between the individual chains in cellulose microfibrils. So here you can see that this is one microfibril, this is another microfibril, and these microfibrils are joined by hydrogen bond. The alternate arrangement of glucose molecule in cellulose also contribute to the high tensile strength of the cellulose. So you might have observed, children, that while building a wall, you might have observed that how the bricks are kept. So in one row, if two bricks are kept side by side, then on the above row, the brick is kept just above the space that is present above the two bricks of the lower row. So exactly here the arrangement is like that. That's why it makes the cell wall more stronger. So this is the cellulose fibril and this is the cellulose structure. Then the next polysaccharide is starch, which is also formed by glucose, and it is the storage form of carbohydrate in plants. And it is synthesized by the most part of vegetable cells and stored especially in seeds like cereals, legumes, tubers, that is potatoes, roots like carrot, radish, and some fruits like green banana, and so many other examples are also there. So this is the structure of starch. So here you can see this is glucose subunit, this is another glucose, and this is the first carbon atom of this glucose, first glucose, and this is the fourth carbon atom of the second glucose. So 1-4- glycosidic linkage is present in starch. Okay, so this starch may be straight chain, okay, and there may be branched chain. So when they are having branched chain, they are known as amylopectin. So when branched chain is there, then that chain, the two sugar units or the glucose units are joined by 1-6- glycosidic linkage. So here I will show you what is exactly 1-6- glycosidic linkage. So here you can see this is the straight chain of glucose where 1-4- glycosidic bond is there. Okay, so this forms the straight chain. But here you can see this is the branch of branch that has been arised from this glucose unit. So here you can see that this is which number of carbon atom. So here from here it starts 1, 2, 3, 4, 5, then 6. So this is the sixth carbon atom and this is the first carbon atom. So the OH of first carbon atom and H of sixth carbon atom is removed as water and 1-6- glycosidic bond is formed. So this is how the side chain is formed by 1-6- glycosidic bond. So this is the picture of amylopectin, which is the branched form of starch, and amylose, which is the simple form of starch where only straight chain is there. Now one of the most important question comes in your exam, children, that why starch reacts with iodine to form starch iodide, whereas cellulose cannot react with iodine. The reason is that starch is having branched structure, and moreover you can see that in both the cases, here it is having branched structure and here it is forming a helical structure. Whatever may be the form of starch, that starch has space or it can hold the iodine molecule in the helix. So here in this picture you can see that these are the branches of the starch molecule, the branched helical structure of the starch molecule, and when iodine is added to the starch solution, then what happens? The iodine molecule is accommodated in the helix of the starch and it forms a starch iodide. That's why the white color of the starch solution is turned into blue-black color. So that's why it forms a starch iodide. But if you look into the structure of cellulose, the cellulose is having straight chain. So here you can see this is having straight chain. So when will it accommodate the iodine? So there is no space to accommodate iodine. So that's why they cannot accommodate iodine and they cannot react with iodine. So this is the important reason that why the iodine cannot react with cellulose but it can react with starch. Very important, children. Then the third type of polysaccharide, third example of polysaccharide is glycogen, and it is the energy storage that is stored food in fungi and animals. The polysaccharide structure of glucose here shows the primary storage form of glucose in the body. Actually, this glycogen is stored, and whenever energy is required, it is broken down into glucose. Then glucose is broken down to release energy. Okay, and in our body, the liver cell stores the glycogen, and the glycogen acts as the secondary long-term energy storage. For long term, the energy is stored. Energy means here the food is stored in the form of glycogen. In muscle also the glycogen is present, and very quickly this glycogen which is present in the muscle is converted into glucose. Okay, and then it releases the glucose, and that glucose is utilized throughout the body. So this is the structure of glycogen. As I have told you, they have both straight chain as well as the side chain. So, in every 10 unit, they have a branch. So, you can see that how they are so profusely branched and their structure is complex. Now, one of the most important thing, children, you have to remember that the glycosidic bonds here shown by that is 1-4- is by one color, and the green color shows the glucose residue linked to by 1-6- linkage. Okay, and one of the important thing here you have to remember that this glycogen or any carbohydrate has two ends. That is, one is reducing end, which is attached, that is which is present on the right side, and on the left side the non-reducing end is there. Okay, non-reducing end means the fourth carbon atom of the glucose, and reducing end means the first carbon atom of the glucose is free. Now let us discuss some of the function of carbohydrate. It is the main source of energy. I have already told you that whatever may be the form of carbohydrate, it is ultimately broken down into glucose. Then the glucose is oxidized to release energy, and that energy is trapped in the form of ATP, and then these ATP are utilized by our cell or body organs whenever necessary. Then reserve food materials are the carbohydrate. I have already told you that glycogen and starch are the reserve food material in different organisms. And when excess glucose is there in the body, then they are converted into fat. So there is a limited amount of glucose which can be converted into glycogen. So if excess glucose will be there, then that will be converted into fat, and that process is called lipogenesis. Besides that, the carbohydrates also helps in formation of the cell organelles and the cell compounds, like the pentose sugar, that is ribose sugar, is the component of RNA, DNA, ATP, NAD, etc. Now the next molecule, that is a biomolecule, macromolecule, is the lipids. So let us see what are lipids. Lipids are organic compounds found in every type of plant and animal cell. And what are the elements? The elements are carbon, hydrogen, oxygen. Okay, but in carbohydrate also carbon, hydrogen, oxygen present. Then what is the difference between these two? In carbohydrate, they are present in the ratio 1:2:1. But here the oxygen, the amount of oxygen is far less than that present in the carbohydrate. And very important children, you have to remember all lipids are insoluble in water. And these lipids perform many important functions, like they contribute to the cell membrane structure. You know that the phospholipoprotein is the chemical nature of the cell membrane. Then some hormones are synthesized from lipid. Then energy storage molecules, the fat stores huge amount of energy. Then when under the skin, beneath the skin, the fat is deposited in the adipose tissue. They help in thermal insulation. Then they also help in mechanical protection. They also help in electrical insulation of nerves, and they also helpful in waterproofing and buoyancy. So this is the structure of a lipid. Rather, I will say this is phospholipid. So you just forget about this phosphate. So here phospholipid is given. So if we will remove this phosphate, then this is the lipid molecule. But the phospholipid is also a type of lipid. So each lipid molecule has two parts. One is hydrophilic head. That means this portion of the lipid can be dissolved or it can be soluble in the water. Okay, but the hydrophobic tail, the tail part consists of the fatty acid which is not soluble in water. So it has two parts, hydrophilic head which consists of the polyhydric alcohol. Okay, and the other one is fatty acid, which may be saturated fatty acid or unsaturated fatty acid, which is not soluble in water. So it is hydrophobic tail. So the hydrophilic head consists of glycerol and it may have phosphate. Now this classification of lipids. The lipids are classified into simple lipids, compound lipids and derived lipids. The simple lipids are esters of fatty acids. They are fats and waxes. So the esters of fatty acids, I will tell you what are esters. I will show you the picture so that it will be clear to you. The esters of fatty acid and glycerol are the fats. The esters of long chain fatty acid and long chain alcohol are the waxes. Then compound lipids are the esters of fatty acids and alcohol, contain other groups also. The derived lipids are composed of hydrocarbon rings and a long hydrocarbon side chain. Now let us discuss each type of lipid one by one. Okay. Before that, I would like to tell you the linkage present in lipid. The lipid is formed by the combination of polyhydric alcohol. So here you can see this is polyhydric alcohol. So here it is glycerol. The simplest polyhydric alcohol is glycerol, which is having 3 hydroxyl group and 3 that is more than 1 hydrogen. So these three OH group are there in the glycerol, and then they can combine with, they can be linked with 3 fatty acid. So fatty acid means the organic compound which is having carboxyl group, that is COOH as the functional group. Remember children, because now you have not studied about the organic chemistry. So let me tell you, this is an organic compound which consists of carbon and hydrogen, and it is having a functional group, that is COOH, that is COOH. It is called carboxylic group. So it is a fatty acid. Okay, so this is another fatty acid. This is another fatty acid. So here you can see that from the fatty acid the H is removed and from the glycerol OH is removed. So there are 3 OH and 3 H from 3 fatty acid are removed. So here you can see 3 water molecules are removed and the bond is formed. So this is the ester bond. This is ester bond. They are formed by ester. So that's why it is known as ester of, what I have said, ester of fatty acids and glycerol. So this is the structure of a fat. So the example is triglyceride. Okay, so this is ester of glycerol and fatty acids. So there are 3 fatty acids and 1 glycerol. Okay, so this is the structure of glycerol. So I think it is clear to you what is ester of fatty acid and glycerol. Now we'll discuss. I have told you that the fatty acid may be saturated or unsaturated. Saturated means when there is no double bond or triple bond, only single bond is present between the carbon atom of the fatty acid, then that is called saturated fatty acid. So here you can see that between the carbon atoms of this fatty acid there is no double or triple bond, only single bond is there, and at the end the COOH group, functional group, is there. So it is saturated fatty acid. Now coming to the unsaturated fatty acid, here you can see in the chain the double bond or triple bond may be there. One or more double bond or triple bond may be there. So in this picture you can see that it is having a acidic group, that is carboxyl group, but here double bond is there. So it is unsaturated fatty acid. Now here are the some of the common examples of saturated and unsaturated fatty acid, and in your textbook only 3 examples are there, but here I have given 3 more. So you have to remember the number of carbon atom present in the fatty acid and the whether they are saturated or unsaturated. So the first one is lyuric acid, that is having 12 carbon atom and it is saturated. Myristic acid having 14 carbon atom and it is saturated. Palmitic acid having 16 carbon atom and saturated. Stearic acid, oleic acid, linoleic acid, linolenic acid, all are having 18 carbon atom, but out of them stearic acid is only saturated and oleic, linoleic and linolenic acid are unsaturated, and the position of double bond is also given here, that is not required for you, that is required for higher classes. Then archidic acid and archidonic acid, these two are having 20 carbon atom, but archidic acid is saturated whereas archidonic acid is unsaturated. Then lignocenic acid is having 24 carbon atom and it is saturated. Now we'll discuss about the difference between neutral lipid and waxes. The waxes are indigestible, whereas neutral lipid, that means I have shown you the fat that can be digested by our digestive system. And in case of wax, the long chain monohydric alcohol and 1 fatty acid. Remember children, in wax only 1 fatty acid but long chain monohydric alcohol is present. That means only 1 alcohol group is present. But in fat that I have shown you the structure, that is a neutral lipid which is having glycerol and 3 fatty acid. So generally in waxes the fatty acids are palmitic acid or stearic acid, but in neutral lipids or fat the short chain fatty acids are generally there. Then acrolein test not required for you, unseatability is also not required for you. And what is the nature of the wax at room temperature, it is a hard solid, and neutral lipid is either liquid or semi-liquid or soft solid. Okay, and waxes have no nutritive value, but the fats or neutral lipids have nutritive value. The example of waxes are bee wax and carnauba wax, whereas the example of neutral lipids are the butters and vegetable oils. Now, what is the compound lipids? So we have discussed the wax and the neutral lipids are the simple lipids which are composed of the that is alcohol, that is polyhydric alcohol or monohydric alcohol along with fatty acid. But what are compound lipids? The compound lipids contains additional substance other than carbon, hydrogen and oxygen. So they may have, along with carbon, hydrogen and oxygen, they may have sulfur, phosphorus, amino group, carbohydrate or proteins. So besides fatty acid and alcohol, they are having sulfur, phosphorus, amino group, carbohydrate or proteins. So the compound lipids are also called conjugated lipid. So the most common type of conjugated lipids are phospholipids when conjugated with phosphate, glycolipids when conjugated with carbohydrate, lipoproteins when conjugated with proteins, sulfolipids when conjugated with sulfate, aminolipids when conjugated with amino acid. So this is one of the example of a compound lipid. So here you can see that this is the triglyceride, that is glycerol and fatty acid. So this is fatty acid and this is glycerol. Okay, here you can see the structure. This is glycerol and this is fatty acid. So other than these two, what else is present? Phosphate group is present. And this phosphate group may be attached to a long alkane or alkene or alkyne group. So this X may be any one of these. Okay. So, but this should have a phosphate group. That's why it is called phospholipid. Then derived lipid. The derived lipids are the substances which are derived from simple and compound lipids by hydrolysis, by breakdown of the simple and compound lipid, that is the derived lipids are formed. It includes the fatty acid, alcohols, monoglycerides, diglyceride and steroids. So this is one example of the derived lipid which is having, as I have told you, that is having ringed structure. So this is the structure of cholesterol, and cholesterol is usually having a ringed structure, that is 4 ring structure and a long side chain, long chain. So these are the long chain. This is the carbon skeleton of a steroid. Okay. So children, today in this session we discussed the 2 macromolecules, that is carbohydrate and lipid, and I hope the structure, the formation of the bond linkage, classification of carbohydrate and lipids are clear to you. I have tried my level best to make you understand each and everything of this topic. So in our next session we will be discussing nucleic acid, metabolic basis for living, that is ATP, then dynamic states of body constituents, then concept of metabolism. So these 3 things we will be discussing in the next session. So children, please go through this video very seriously and carefully listen to my explanation so that your concept will be clear. And again I am telling you children, these concepts and these understanding of the formation of bond, the structure, the classification are very, very important. Okay. So thank you children for watching the video till end. Thank you once again. Thank you.